I have a question for you. Y'all ready for the question? Why did the detective stay in bed? Why did the detective stay in bed? He had the case of a cold. Okay, it's a good guess. He wanted to stay undercover. Yep. It's a good answer. What do you call a fake noodle? An impasta. Nicely done. Well done. You've heard that one before. What did the brother Sel say to his sister when she stepped on his toe? My toe, sis. Yeah, come on. That's a tough crowd. I get some giggles. Some of you still don't get it? Okay, that's a problem. That is a total dad nerd joke right there. Quiz 12, due Sunday by 11:59. Don't wait till Sunday. I tried hard last week to remind you. Even on Easter, I sent out reminders. Do you guys remember that? About the 2 quizzes? So just don't wait until Sunday and then forget. So do it early. It's on chapter 12, Muscle Physiology. And then did you see that the study guides posted for the exam? I had to make sure that I felt like we were going to cover everything before I put everything up there, but I'm feeling pretty good about it. Okay? So the study guide's up there. There's going to be some more SI sessions. There's going to be a review session. The TAs and SIs and I met yesterday, so we're going to kind of do that same theme this weekend. Okay? I have one more office hour tomorrow. I told you it was by Zoom, which is true, but I'll also be in my office in Wetah, 1:30 to 2:30. There was one student that forced me to change it to in-person. Not forced, that's the wrong word. Requested. I don't know where you are, but it's for you, wherever you are. I can't find you. So if others need to come in person, 1:30 to 2:30 in Wetah, I'll be there. We're going to start lecture with this video because I want you to be able to visualize in motion how this works. You get about 5 power strokes per second. Okay, 5 power strokes per second. And in order to appreciate what that looks like, I think a video does it the best. And it's got this really great music.
We move our bodies by contracting skeletal muscle, while the results of this action are plain to see. The mechanism behind the action is not. Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended into what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, Adenine triphosphate molecules attach themselves to myosin heads. These molecules trigger the release of myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle.
So watch this motion right here. Oh, they already showed it. I thought they were going to show it again. Hang on.
Muscle tissue occurs when actin and myosin myofilaments slide past one another, causing.
Do you guys see how those power strokes move?
It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs.
So is that clear? That make a little bit more sense? How you've got these interactions between the myosin head and the actin, and in the presence of ATP, in the presence of energy, you're going to be able to cycle the cleavage of the phosphate groups, which is really important to remember. And we're gonna talk about that in more detail today as we move on. Hang on, I'm just getting something organized here. Bear with me. And I wanted to start the lecture with a video because some of you in office hours yesterday were having a difficult time conceptualizing, which is totally understandable. I mean, this is a pretty unique concept, and it's not like really anything you've ever seen. And so I feel like having the opportunity to see it in motion, which is hard to do with still pictures, you just have to get back kind of the old school elevator music and the monotone voice, but they do a really good job of demonstrating, illustrating, and that's a really old video. I mean, it's been on the web for a long time. Question?
If you're holding your muscle in a flex, is it still making those strokes, or is it just staying at the fifth one?
Great question. If you're holding your muscle in a flexed position, is it making the strokes, or is it just attached? It's just attached. Okay? The cycling is actually burning ATP. And in fact, you need ATP for the detachment to take place. We're going to talk about this today. So when you have the attachment and then you fire, you kind of have the boom, 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 right? Every time it detaches is when ATP binds to let go. When no more ATP is available, the muscle stays in a contracted state called a rigor complex. It's actually referred to as rigor mortis. And that's what is a stiff on forensic analysis, right? Someone shows up, the individual's been dead for a while, and their muscles are stiff because all of the actin-myosin crossbridge formations are still attached. They haven't let go because there's no more recycling of ATP. Make sense? We'll talk about that here in a second. Here and then here. No, that's called the rigor complex, and rigor mortis is basically when ATP is not available anymore. Yep.
What happens when you cramp? I mean, there's like muscle flexion, there's just stuck there? Or
Yeah. So the question is on cramping. So there's a couple things that could be happening with cramping. One is you could have a sustained action potential that isn't repolarizing or relaxing, and a lot of that's due to potassium inavailability, right? Because potassium is responsible for the repolarization side. Secondly, it could be lack of availability of ATP in that moment in time. And third, it could be you're transitioning from one source of energy to another, which We might get in the end of the lecture. If not, we'll cover it on Monday. Okay? Good questions. Let's dive into this in a little bit more detail from where we left off. I cut my finger, so I'm trying to make sure I don't bleed all over my shirt. Relaxed versus contracted. If you took a look at these two images, you should be able to tell the difference between a relaxed state and a contracted state. Now, you tell me, how do you know that this is relaxed and this is contracted? And the answer isn't because I labeled it. The Z lines are closer together in the contracted state. Perfect. The M line doesn't move. And the concept is that those filaments, like you saw in the video, are going to slide over top of each other. So we've got a couple of different states that we can look at. We can look at a relaxed state, which is shown up at the top in this schematic. Here's the corresponding electron micrograph. And you can appreciate that the thick and the thin filaments don't change in length as we go down, but as it's contracting, here we see sort of a maximal amount of overlap, if you will. And then here we see that the thin filaments are kind of crowding over top of each other, and it actually is in a fully contracted state when you've reached kind of the end of the thick filaments. You with me? So there's definitely a range at which you can contract the muscle. And we'll look here at a little bit of length-tension relationship a little later in the lecture, but I just want you to understand this concept. We're going to go through this very generally, because in the next set of lectures that we're going to get into right away, remember this is the very end of the last lecture, we're going to go into the step by step by step. So I'm going to do an overview, and then I'm going to switch the slide deck. I'm not even going to pause the video, and we're going to roll into the next set of lecture. Okay? But the concept is right here at the neuromuscular junction. We've got the nerve coming down. It almost is like it's reaching its hand out to touch the muscle cell, the muscle fiber. And we looked at this in great detail in the last lecture, and we kind of characterized all the different attributes. One thing I want to show that became a little bit more obvious in office hours yesterday was someone asked me the question what we were looking at, and I failed to point out from this slide. Do you see these little holes? They might be hard to appreciate from your seat, but if you look at your picture, there are these little holes. This is the plasma membrane of the muscle cell, sarcolemma. The action potential is going to come down the membrane. Remember, this is the phospholipid bilayer. And it's going to dive into that channel, and that's what this middle T-tubule is. It's called a transverse tubule because it's a tube that runs from the outside into the center in a transverse cut. Because if you're this bundle right here, you're not anywhere near the membrane. So you need the action potential to dive all the way deep into, to have the full access of all the actin and myosin within the cell. That's the function of these T-tubules. And you can see there's holes all throughout the membrane. You can also appreciate that the sarcoplasmic reticulum in blue is peppered throughout the entire cell. It surrounds every one of these myofibrils. And I didn't really highlight that, so I want to make sure, because I think that will help with today's lecture as we look at excitation-contraction coupling. Does that make more sense? Okay. So, on this slide, whether it came from the sarcolemma and it's near one of the myofibrils, or if it's deep in the center of the cell and it came down the T-tubule, the transverse tubule, you're ultimately getting an action potential that is interfacing with the cell, stimulating the release of acetylcholine, and it's the sodium and potassium propagation that takes place that stimulates a voltage-sensitive calcium channel to allow calcium to flood into this end of the synaptic knob. That flood of calcium stimulates acetylcholine to release its content, fuse with the membrane and dump its contents. Now, acetylcholine is going to bind on the other side of the synapse to its receptor, and it's going to open a channel and allow sodium to come in and potassium to go out. And if sodium comes in, that depolarizes the muscle. Make sense? So this is excitation, contraction, coupling, beginning. And down here you can kind of see once this happens, now you stimulate these voltage gated ion channels to open respectively, just like we learned in the last unit. Now, what about the coupling part? Well, if we look at this picture, which is what I was just trying to describe, we have the membrane, the sarcolemma, and if the action potential propagates down and then dives into the depths of the cell down the T-tubule, on either side of the T-tubule are two components of the sarcoplasmic reticulum that stores calcium in the muscle cell. That action potential signal will trigger calcium to be released out of the sarcoplasmic reticulum, move into the cytoplasm of the cell, the muscle cell. Calcium can find troponin. It binds. As it binds, it causes a shift in the shape. It's like if you pick up, a heavy bag, all of a sudden your position changes. You're not walking, all happy-go-lucky. You're carrying a big old bag and you're kind of like, so you go through a three-dimensional change when you grab something. That's exactly what troponin does. There's a three-dimensional shift because calcium binds. Because tropomyosin is attached to calcium or to troponin, it moves with troponin when troponin binds calcium. Remember, these are two proteins we refer to as regulatory proteins because they regulate whether or not myosin head will find the binding site on the actin molecule. And that calcium release from the sarcoplasmic reticulum or from the terminal cisternae of the sarcoplasmic reticulum is critical for tropomyosin to move out of the way, exposing the binding site so the head can find the actin molecule and do the power stroke that you just saw in the video. Make sense? So a couple of terms as we back up. But this concept is what we refer to as excitation, contraction, coupling, because you've just taken an electrical signal and we've just linked it to muscle contraction. Excitation, contraction, coupling. Some terminology. The motor end plate. The motor end plate is where the acetylcholine binds to trigger an action potential on the muscle sarcolemma. That's the motor end plate. Why is it called a motor end plate? Well, it's a motor function because it's a muscle. It's an end plate because in electrophysiology or in electrical currents, you refer to the end plate as where the electricity goes. Okay? In the literature, it's also referred to as a end plate potential, an EPP, like if you read some other papers. But that's what motor end plate refers to as the end plate potential or the electrical signal that's happening on the muscle side. The synaptic cleft should be pretty obvious. That's that gap between the neuron and the muscle. Acetylcholine, we've talked about, is being released out of the synaptic knob. And then acetylcholine esterase. This is an enzyme that's released into the cleft to break down acetylcholine into its constitutive parts, acetyl and choline. And then it's reabsorbed on the presynaptic side and recycled. So if we look at the coupling event, this is the contraction part. This is part of what the video was showing, and I'm showing it in still photography, right, or still animation. So I want to start right here. I know this is labeled 13 and this is labeled 10, but I want to start right here because in this format, you've got a full molecule of adenosine triphosphate. How many phosphates does adenosine triphosphate have? 3. Well done. Okay. That was Unit 1 material, right? In this state, you can see the orientation of the myosin head. And when you move from this state to this state, what do you notice about the position of the head? It goes from here to here. So it's like cocking the hammer on a gun, right? So you cock it back. Well, in order to do that, you cleave one of the phosphates, and you're left with adenosine diphosphate, which is 2, plus a free phosphate that just hangs out here. So it takes energy to cock the myosin head. In this position, though, it is available to bind. It'll find the binding site, assuming that calcium is present, and this has the tropomyosin has moved out of the way because troponin has bound calcium. Tropomyosin is attached to troponin, and when troponin has a conformational change, tropomyosin moves with it, exposing the binding site. Now with a cocked myosin head, it can find the binding site and form what we call a cross bridge. That is the cross bridge right there. Actin and myosin stuck together. Okay? On the head here, remember we've got adenosine diphosphate in one phosphate group that's kind of already been cut. And in order for it to go from a cocked position to a fired position, you cleave those extra 2 phosphates. You burn all the energy here in this power stroke. And then to let go, detach, you add another new adenosine triphosphate, it detaches, and you're starting in this position. And that's the cycle. Does that make sense? Questions over the cycle. So myosin attaches to what? The myosin binding site where? On the actin filament. Well done. We went through cocking, attachment, power stroke, reattachment. Now let's talk about relaxation. So when you're done with this cycle, how do you relax the muscle? Well, you release acetylcholine, esterase, to digest the acetylcholine. You what we call resequester calcium. So active transports in the sarcoplasmic reticulum. Remember, that's just a modified endoplasmic reticulum inside the cell. It has active transports. So these are pumps. If it's an active transport, does it require energy? Yes. Let me rephrase that, because that would be a great way to ask a review-style question on the next exam that's relevant to this material. But it's also relevant to Unit 1. The active transport of calcium back into the sarcoplasmic reticulum, does it require energy? Yes. What kind of energy does it require? ATP is our favorite kind of energy, right? Shouldn't be a surprise. That actively pumps it back in to the sarcoplasmic reticulum. Inside there, it binds to a molecule known as calsequestrin, which has a high affinity for calcium. And so it'll grab the calcium that's coming in to the sarcoplasmic reticulum. And it's stored. They're like grain silos. If you've ever driven across the country, or if you're from the Midwest, you've been to the Midwest, you drive and you just see like tons of giant grain silos storing all this grain. Think of the sarcoplasmic reticulum and calsequestrin as like a storage house for calcium. But it's local inside the muscle. It requires ATP. ATP is required for the myosin head to detach as well. And we talked about rigor mortis. I already mentioned it. Bless you. This starts 3 to 4 hours after death. It peaks at about 12 hours. And then eventually it dissipates, meaning the tissue decomposes. And so once decomposition takes place, the muscle starts to just break down. So the dissipation or the decomposition is about 48 to 60 hours is when you're going to get detachment. So if you hit it in that window of 4 hours to 12 hours, you're going to have a very stiff cadaver, okay? And the problem with rigor is you don't have ATP being made, so you don't detach your myosin head, and you can't get rid of calcium because you need an active pump in order to do that. So this is referred to as the hardening of muscles or the stiffening of the body after death. Post-mortem is the name for that. But eventually, you get decomposition of the tissue, and the tissue will become more pliable again. So, take-home points. Muscle relaxation requires ATP. And if ATP production is no longer generated, you can't relax the muscle, which was one of the questions over here on cramps, right? So the question early on about cramping is, it could be an ATP issue, right? You've just burned all of your ATP. So the fibers ultimately decay and things kind of decompose, yeah.
So why after something's just been killed, if you put salt on it or something, might the limbs still move?
Well, the rigor complex isn't going to happen right away. It's 3 to 4 hours post-mortem is when it's really kind of setting in. And so, if there's any hunters in the group, you know this pretty well. And if you field dress your hunt pretty quickly, you really don't have any problem. But typically by kind of the next morning, the meat is actually much firmer than it was when it was, freshly taken down. So the salt typically helps with drying out the meat, and that's valuable because it serves as a preservative, right? And if it's too moist for too long or has any bacteria that is contaminated, and salt will actually have the tendency to kind of kill some of the bacteria that might set in. But it won't really prevent rigor from taking place.
Is there ever a point where it can no longer be reused, or is it just able to be used as many times?
Yeah, so the question is, when calcium is stored, is there ever a point at which it becomes useless or it can't be recycled? I don't think so. It's a pretty resilient molecule. I would imagine it may have a lifespan, but again, you have a pretty homeostatic control over your calcium levels. So if your muscle is low on calcium, you'll actually stimulate osteoclast activity, break down a little bone, free up new calcium, bring it into the bloodstream, bring it into the muscle and store it, right? And you'll do that before you'll have too little calcium to do muscle contraction. But you have to remember, most of us are thinking skeletal muscle. Smooth, cardiac muscle also require calcium for contraction. And so calcium is something you don't want to be short of, right? You'll stop breathing. Your heart won't pump anymore. So in patients where you have to choose between, calcium availability and having an appropriate bone mineral density, you can see why you'll steal calcium from the bones all the time, right? Your body will. Good questions. We watched this video, but the link is here so you can rewatch it if you just need to see the picture one more time. So let's answer a couple of questions. What surrounds each muscle fasciculus, and in contrast, what surrounds each muscle fiber? So this is a microanatomy question. What surrounds each muscle fasciculus? And that's the first blank. What surrounds each muscle fiber? Any ideas? You look back at your figure. No volunteers? Okay, this might be a good study one. So the paramycium surrounds the fasciculus, and then the endomycium surrounds each fiber. And we talked about that in the first muscle physiology lecture, just a microanatomy type of question. Okay, let's try this one. The enlarged areas of the SR, the sarcoplasmic reticulum, near the T-tubules are called what? The enlarged areas of the SR, the sarcoplasmic reticulum, they're near or adjacent to the T-tubules. What are they called? Terminal cisternae. Terminal cisternae. Well done. Questions?
So if breaking myosin away from actin requires ATP, why does maintaining a muscle contraction take so much energy? Like wall stints, why does that cause you to feel tired if you're not breaking those bonds?
Yeah. It's a great question. So I'm going to repeat the question so you can hear it, and also it's on the video. If actin and myosin interaction requires ATP, right? Why then does maintaining a contraction require so much energy, or why is it so difficult to do? Okay, are you recruiting more ATP? What's happening there? So it comes to an issue of recruitment, and we're going to get to it here in a little bit. But in order to do an activity, your body is going to make sure that what happened? Okay, that just scared me because we really need the video to continue. I think it just went to sleep, I'm hoping. Okay, so your body's going to do things as efficiently as it can. And so if you can do an activity, as you get more skilled at the activity, you're actually going to use fewer muscle fibers to perform that activity. Okay? And when you start becoming fatigued, you're going to recruit additional muscle fibers based on recruiting additional motor units, which is the motor neuron and the muscle fiber that it innervates. That's a motor unit. You remember that definition? So if let's say you're doing an activity where you're throwing a ball, right? Or actually, a better one is you're doing an activity where you're holding this up, right? You're holding this up. And you're going to hold this up for a long period of time. And let's just pretend it's solid concrete, and it's 7 times as big for the demonstration purposes. And I'm holding it like this, and the trainer's telling you, hold it as far out in front of you as possible. Or after about an hour, right, I'm going to start shaking because I'm recruiting new motor units to do the same activity to hold the position. And in the beginning, I used very few. And as I become fatigued, I recruit additional muscle fibers by recruiting additional motor neurons. So motor units are being recruited to maintain the position. Does that help? And when you do that, you actually have to recruit additional ATP to hold that position. So we'll talk about isotonic and isometric exercises. I don't know if we'll get to it today. We might be on Monday. But it's a great question, so stay tuned. Here's some homework for you, and then I'll get to your question. I want you to draw these out. It doesn't have to be pretty. I really think, in my opinion, it would be wise for you to draw this on the exam packet so that you have something to go to when you're stuck and you're like, I'm so confused right now. I don't know what I know. I don't know what I don't know. Okay? And you're mumbling swear words about me under your breath. Just draw this out because you know a lot of information. And then refer back to it on your exam packet. There's plenty of white space or empty space on the packets to draw. So practice doing this and know these different structures. It doesn't need to look like this, but you can make a thick, dark filament. You can make a thin filament. You can draw kind of a corkscrew, thin filament. Put in your Z-disc. I would start with the Z-discs because it defines what it is, a M line down the middle. Just get familiar with what's moving across what. Okay? So I really encourage you to work on that. It's probably an activity you'll do in SI or in the review sessions, and I think you'll find it helpful. Okay, the specific part of the sarcomere structure that's only occupied by the thick filament and is lighter in color than the rest of the particular band is called what? And this is a perfect example. If you're freaking out, you go to your drawing. You're like, I don't know, I don't remember. I thought I remembered this stuff. H zone. Well done. Okay, so it seems like some of you are already studying this, which is great, because the exam's a week from today. Is that a surprise, seriously? Okay. No, the exam's a week from today. Seriously? Do you all have a copy of the syllabus? You guys remember this? The exam's a week from today. It's not Monday. Were you thinking it was Monday? Yeah, a week from today. So much changes these days. I don't know. I mean, I'm not on social media, so maybe it's a different way to describe the weeks now. I don't know. It wouldn't surprise me. Okay, exam is, well, I'm glad I mentioned it. Exam is a week from today. So we're gonna get pretty far today, and we'll finish up on Monday, and we might be able to do a little review on Monday, like we did last time. Okay? All right. Now that we got that all cleared up, I'm glad we had that conversation. Oh, I shouldn't have said anything. You would have come Monday, like, ready to take the exam, and then I've been like, I just punked you all. It's Wednesday. I know that's an old term. I'm showing my age. Muscle physiology. Let's talk about excitation, contraction, coupling. We're going to just do it again. We're going to walk through this concept one more time. Excitation, contraction, coupling. And we're going to take it one picture at a time. The pictures will be bigger. We'll go step by step. There's 4 main phases. 4 main phases. I could see a question like, which of the following is not a phase of excitation, contraction, coupling? Right? I mean, that's an easy. There's 4 phases, and I could put one that doesn't make any sense. You have excitation. You have the process where the nerve potentials lead to a muscle potential. You understand what that terminology means? A nerve potential leading to a muscle potential. We use very specific nomenclature in this class that doesn't translate except in the field. And some of you are telling me, Keller, I just have trouble understanding what your question's asking because you word it so weird. I'm like, yeah, there's a lot of Latin, a lot of Greek, and a little bit of English in every test question. Awesome. Welcome to the field. So, action potential. You've got a nerve action potential that leads to a muscle action potential. Excitation, contraction, coupling. These are the events that link the action potentials on the sarcolemma to the activation of the myofilaments, getting them ready to contract. What are the myofilaments? What are the names of the myofilaments I'm referring to? Actin and myosin. Okay. Contraction. This is the step in which the muscle fiber develops tension and may shorten because you get the Z discs moving closer together. And then last, but very important, is you actually need it to relax and return to its resting state. So excitation, let's take it one step at a time. Excitation. This picture should look familiar now. You've got step number 1, arrival of a nerve signal, which we studied in the last unit. We went over this. We had graded potentials that would summate, they would reach threshold, which is minus 55, then an action potential would fire, right? Resting membrane potential was about minus 70. You remember all that? Just kind of vaguely, you remember that? I'm not asking questions about those specific numbers in this unit, okay? But I'm just trying to recall and put it together. So the nerve signal opens voltage gadolinium calcium channels at the synaptic knob. Calcium, so the voltage signal comes down here, a calcium channel opens, calcium comes in, and this actually stimulates these vesicles to dump acetylcholine into the synapse. Kind of step number one. This little box right here is blown up in higher magnification so you can see it. The little green spheres are the artist's representation of acetylcholine molecules binding the acetylcholine receptor on the postsynaptic side, the motor end plate. If we take a look at what's happening to the receptor, acetylcholine molecules, 2 molecules of acetylcholine bind to its receptor. It goes through a conformational change, opens a channel, allows sodium to flow in and potassium to flow out. Review question. What establishes high sodium outside the cell and high potassium inside the cell? Or what helps to maintain it? Sodium-potassium pump. Well done. Okay, that's review. We talked about it in Unit 1. We talked about it in Unit 3. We're using it in Unit 4. It will never go away, folks. You will always have to know the sodium-potassium pump. Okay? It's a really important pump, if you can see that. So now sodium comes in, depolarizes the inside of what cell? Where am I? The muscle cell, also known as a muscle fiber. Well done. And what membrane is this? It's not a sarcomere, it's a sarcolemma. Sarcolemma, like, yeah, that one, Keller. That's sarco. This is the sarcolemma, which is a plasma membrane, a phospholipid bilayer, right? Again, that's Unit 1 stuff. We talked about it in Unit 3 because that was nerve physiology. We're doing it again in Unit 4 with muscle. Do you see all these motifs that continue to build? That's why many of you are like, man, the first part of this class is so hard. Yeah, because it's like learning a foreign language. Now you're starting to apply a lot of the stuff. And guess what? Next semester in 202, you'll apply a lot of this again in 202. Okay. Now, do you see this picture? Here's where the knob was. Here's the cleft. Here's the acetylcholine receptor. Here's our 2 acetylcholines. Now we send this motor end plate or an end plate potential, a voltage change in the muscle membrane, and it's propagating by opening the respective voltage-gated ion channels, just like we talked about with nerve physiology. Nothing new. Same kind of channels. Those channels worked well in the nerve. The body says, I'm going to use the same exact tire. I'm going to make it round. I'm going to put air in it, and it's going to work really well. It makes sense? So this same motif of a sodium voltage-gated channel is bringing sodium in. That's propagating the action potential down the sarcolemma. You follow? So now we go down the sarcolemma and we come to this hole in the membrane that has a tube that goes all the way into the belly of the cell because there's myofibrils in the middle of that cell that I need to get the action potential to. And so it dives down that T-tubule. Make sense? Conveniently located on either side of the T-tubule are bags of calcium inside the cell. We're on the inside of the cell, though. Right? So this T-tubule is still kind of outside the cell. You with me? Inside the cell, just on the other side of the T-tubule membrane, is where those bags of calcium are. Those action potentials that spread down the T-tubules, they trigger, amazingly enough, voltage-gated channels. You're like, oh, I think I'm starting to get it, Dr. Keller. Is it starting to kind of click? You're using some of the same old stuff. Voltage-gated channels in the T-tubules open up channels in the sarcoplasmic reticulum to release calcium into the inside of the cell. Now keep in mind, we've got calcium moving in 2 spots. Backing up, we have calcium moving here at the synaptic knob to release acetylcholine. Now we have calcium moving out of the sarcoplasmic reticulum into the muscle cell. You with me? So you're using calcium in 2 different locations here. Don't get confused by that. Calcium enters the cytosol of the cell. Cytosol and cytoplasm are very similar. The cytoplasm contains the cytosol as well as insoluble components. So the cytosol is the liquid part of the cytoplasm. It's just cytoplasm and cytosol are really almost interchangeable for the most part. Now, where we already talked about in the last lecture, we get calcium binding to troponin. Troponin binds calcium, and it goes through a conformational change. It's like picking up a really big bag of something. Like big, you have two big suitcases. You pick them up. You're not walking, straight upright. You're kind of bent over, kind of clunky, right? So that's the three-dimensional change. It picks up calcium. It shifts in its three-dimensional shape, and it tugs on tropomyosin. And some of you might ask, how do I remember which one's which? The tropomyosin hides the binding site that myosin has to find. Tropomyosin hides the binding site that myosin has to find. Troponin doesn't have myosin in the name. Troponin binds calcium. Now, tropomyosin and troponin have moved. You expose these nice, elegant binding sites for the myosin head, and you get caulking of the myosin head by cleaving ATP. So it doesn't show it, but a full ATP molecule is here. And on this one, it's ADP plus 1-phosphate, inorganic phosphate. So you've already cleaved the ATP to get the head to move back. This is hydrolysis of ATP. That's the terminology. Hydrolysis of ATP is where you lyse the bond that phosphate has and you liberate some energy. You with me? So hydrolysis of ATP is a terminology that means you break one of the phosphate bonds, and from adenosine triphosphate, you go to adenosine diphosphate plus an inorganic phosphate that's available, kind of hanging out. You can see the little shadow of the head here, and now it's moving per the arrow to a cocked position where it can actually interact with the binding site and form what we call a cross bridge. So the cross bridge has to have a myosin in its cocked position to be able to interact with the binding site.
Does the head only cock after the tropomyosin moves out of the way?
Yes. The question is, does the head only cock back after the tropomyosin moves out of the way? And the answer is yes. You'd be burning a lot of energy if you weren't able to bind. You want to keep it in a resting position, and you don't want to burn your ATP if you don't have to. What tells it? Other signals in the molecule with the tropomyosin send secondary signals that you'll learn about probably later in your coursework that stimulate enzyme activity for hydrolysis to take place and the cocking of the head. Now you get cross-bridge formation. Now we move from the bottom here. We're going to start here on the bottom right and move up. You actually get the power stroke. So some terminology. Cross-bridge formation, power stroke is when you fire the head of myosin, like you saw in the video, and that burns up the remaining ADP that's bound. And it's still attached. Notice, after it fires, it's still attached. What is necessary for it to detach? What's that? What's necessary for the myosin head to detach from the active molecule after a power stroke? ATP binding again to the myosin head will release it out of the actin attachment to the actin molecule. Okay, ATP shown right here. So this process happens 5 strokes per second. It's pretty quick. And every single head can be bound through a cross-bridge formation and going through power stroke on an individual fiber. Right? And then if you think about the organization of the muscle, per the question that came earlier, is what if you're holding a contraction Can you recruit additional muscles to do this? Yes. But you're going to start small, conserve your energy, and then add as you need, right? And that's all part of training. Each power stroke utilizes one full molecule of adenosine triphosphate. These are some good statistics to remember because it's a very energy-demanding process. But then, what's an architectural characteristic of the muscle that you know about that makes you unconcerned about how much energy is necessary? What is a organelle architectural componentry of the muscle cell? Has a lot of mitochondria. Does that make sense? Questions?
If you're doing a movement that takes less than a second to do, does that mean you're not using five strokes?
So the question is, if you're doing a movement that takes less than a second, are you not using 5 power strokes? It's an average number, but yeah, you wouldn't necessarily need that many power strokes. With excitation-contraction coupling is always a 2-to-1 ratio, meaning 2 acetylcholine molecules binding to its receptor, yes. So this is the most difficult part of muscle physiology. You compare this to nerve physiology. I think this is more straightforward, but it's also not a fair comparison because a lot of the stuff we already learned, you're applying here. Does that make sense? You're kind of building on what you already know. We're using some of the same components, like with the voltage-gated sodium and potassium channels. Now let's look at relaxation. How do you unwind this whole thing? And aren't you glad you don't have to think about all these steps when you want to walk to the bathroom? I mean, it's pretty amazing that we do this without even thinking about it. So you stop the signal of the nerve. You stop motor efferent signaling. So that stops acetylcholine release. And then you release acetylcholine esterase, that's the ACHE, and that binds to acetylcholine and breaks down acetylcholine. And it breaks it down into its constitutive parts of acetyl and choline, and that's reabsorbed back into the synaptic knob and recycled to make new acetylcholine. So now the stimulation from acetylcholine stops, and you don't send the signal any further and propagate it. So you turn off the signal, essentially. Now, additionally, you resequester is the name of it. You resequester calcium out of the muscle cytosol, and you pump it back into the sarcoplasmic reticulum. So it's coming off of troponin. And then tropomyosin slides back over the binding site for myosin, the myosin head, on the actin molecule, and you won't get cross-bridge formation anymore. This is an active transport mechanism, so it needs ATP to do that. And it wants to go in there because there's a molecule that it has a high affinity for, which is called calsequestrin. So you have 2 forces moving calcium very quickly back into the sarcoplasmic reticulum. You've got calsequestrin, which is kind of, I think it's a cool name, actually, calsequestrin, because it's kind of, I don't know. If I had a son, I was thinking about calling him calsequestrin, but no, what? Seriously? You didn't like? Good news is I had all girls, so there's no calsequestrin Keller. But I think it's a cool name because it sequesters calcium. It's pretty easy. So that's a force that's drawing calcium back into the SR, and you're actively pumping calcium back into the SR. Okay? That's a goofy detail about me, but you probably will never forget it now, right? And you might laugh years later, which would be funny to see happen. So ATP is now needed for muscle contraction as well as muscle relaxation. Calcium's removed from the troponin. It's pumped back into the sarcoplasmic reticulum. Tropomyosin reblocks the active sites. The muscle fiber ceases to produce the tension, and it returns to its original resting length because of its elastic recoil due to what filament? Titin, otherwise known as elastic filament. Well done. And that's the whole cycle. That's how muscles contract. I think it's kind of cool. Questions? So one force, so I said there's two essential forces bringing calcium out of the cytosol of the muscle and back into the sarcoplasmic reticulum. One is active transport of calcium. It's actively being pumped into the sarcoplasmic reticulum. Number two is calsequestrin is housed inside the SR. So as soon as it's pumped in, it wants to bind to calsequestrin. Yeah, so the question is it 100% effective for bringing all the calcium back? I would say probably not 100%, but it's rather efficient, like in the 90 percentage range, 95 to 97% effective, where you have very limited calcium, free calcium floating around in the cell. But if you think about, even if you reduced it by 90 to 95% or 97%, the number of troponin molecules in a muscle fiber is very high. And if you're dropping the calcium concentration so much, you don't need to get to zero for the muscle to relax. Okay? And you don't really want to get to zero because you want some basal level muscle tone of the muscle. All of our muscles have sort of a basal tone. So the question is, if you cleave a phosphate group through hydrolysis of ATP to get adenosine diphosphate and PI, and that causes you to caulk the myosin head, right? Then you burn the rest of that energy. Why do you need a full ATP molecule to relax? Is that the question? You need a full ATP molecule to bind to the head of myosin, because when it binds, then it changes its shape, its conformational shape, and that allows it to no longer fit in the binding site. So it's like it needs to grab onto ATP, so it has to let go of the actin molecule. Make sense? Okay. We're going to continue to go because the more we get done today, we have about 17 minutes, the less we need to cover on Monday, and the more we'll have available for review. Sound good? Study guide's already posted. You're going to want to stick around today to the end because I'm going to give you your bonus opportunity. We're going to get into some of the questions that were being asked in the beginning on some of the dynamics of how this works. So I alluded to the length-tension relationship. And really what this refers to as is trying to maximize the overlap between actin and myosin. So if you look at this graph, you've got sarcomere length on the x-axis, and then you have the amount of tension on the y-axis. And you can sort of appreciate that at some middle range, it has maximum amount of tension that's generated. And this is the picture that's shown where you've got maximum amount of myosin heads able to interact with actin. Here, the muscle is overstretched. And you'll have some interaction right here at the end, but you won't have as much interaction as here. Therefore, you can't generate as much tension or force. Here, they're actually too crowded. And so the best way to kind of exemplify this is, like, in the weight room or in activities, the middle range of the flexion is typically where you're going to get max force production. The middle range of the flexion motion of the muscle is where you're going to maximize the amount of overlap between myosin and actin, and you're going to get the most amount of force. And then if you're, overstretched or overcontracted, you'll generate less force than you will right in the maximum overlap range. So this is referred to as the length-tension relationship. And there's another, hang on, where am I? There's another term that we should go over, which is the cross-sectional area. So the force that can be generated is proportional to the cross-sectional area. So the larger the muscle cross-sectional area, the larger the force production. Because the larger the cross-sectional area of the muscle, the more myofibrils you have. The more myofibrils you have packed in there, the more cross-bridge formation that you get. So bigger muscles generate more force. You're like, well, that seems pretty obvious, Keller. Okay, but this is why. Okay? The central nervous system, bless you, maintains a basal level of muscle tone, kind of to that earlier question we were talking about with calcium, which is one of the reasons it's not 100% efficient. The central nervous system autonomically will maintain a basal level of muscle tone in a muscle so that it is poised and ready to actually contract. And so examples of this would be, we see in stroke patients, we see this in spinal cord injury patients where that central nervous system connection is severed and there's no basal tone to the muscle. Okay, so the muscle is flaccid. It won't contract at all, and the basal amount of tone or the basal amount of cross-bridge formation that's actually happening in the muscle is gone.
Is the basal tone what causes things like wrinkle formation?
Yeah, the basal tone does contribute to wrinkle formation as you get older. It's because all of us have basal tone right now, but in all of you, because you're young, the tissue on top of your muscle is more hydrated, it's more elastic, it's more pliable, so it hides a lot of those wrinkles. As the elastin component of the skin deteriorates as we age and the skin thins out, the basal tone becomes more apparent. Another name for it is tonus, T-O-N-U-S, and that's what we're referring to as this basal tone. So let's talk about how muscles contract with varying strengths, which is kind of one of the questions that we had in the early part of the lecture that I put off to the end, and I was hoping we were going to get to it, and it looks like we are. So here we've got a muscle contraction, or a signal for muscle contraction we refer to as a twitch, a muscle twitch. And we have the ability to recruit muscle activation based upon spatial or temporal. That terminology should sound familiar. If it's spatial, it's what I'm showing right here, directly below, where if you look at these hash lines, these are actually a stimulus, a voltage stimulus. Right? That's our nomenclature with electrophysiology that we saw in the nerve physiology section. It's the same for the muscle. We've got 9 different activations. Here in cross-section, you can see a nerve, and we can see the stimulus of the fibers. As you move to the right, all 9 are being activated. Here, we've got fewer, so they recruit spatially different nerves to fire, sending signals to different muscle groups to recruit them to contract. Okay? And here's the amount of tension on this slide, and here's the number of nerves that we're recruiting in order to get the different maximum contraction events. Now, to the question earlier, you could get up to 7. You can get maximum tension with 7, but if you need to hold it for another 10 minutes or another 15 minutes, this is where you start recruiting additional nerve signals for additional motor units, because a motor unit is the nerve and the muscle fibers that it innervates. And so you can recruit more of those to a certain point to maintain that maximum contraction until you fatigue. Now, another way we can actually summate or add strength on contractions is using temporal summation. And so if you look at an individual muscle firing, so here's a action potential with a muscle twitch. Every one of these is an action potential that led to a muscle contraction. And you can see on trepe is where we can produce this staircase phenomenon, where we can actually add and accelerate the amount of force that's being produced by bringing those twitches closer together. And then we can get to two different conditions. You can see the hashed lines on the bottom are showing like action potential, muscle twitch. Action potential, you see the black little hash down here, and then the red twitch. Over here, you can see that there's more nerves firing, and so you have more muscle twitches. You with me? Here, we can go through what's called incomplete tetanus, where we can bring these closer together, where we can staircase this, like you did in trepe, where you raise the amount of force production that happens until you reach some maximum cramp mode, if you will, muscle cramp, where you get complete tetanus and then fatigue. Okay? So just different ways that we can vary the strength based upon spatial or temporal. So let's finish that story on isotonic versus isometric contraction. So contraction is force over a length. That's typically what it is. If it's isometric, iso meaning same, metric referring to length, so same length, you're generating on an isometric contraction force or tension without changing the length of the muscle. Isometric. In contrast, an isotonic contraction, iso referring to same, you're generating the same force, and you could either have a concentric or an eccentric contraction. So here an isotonic concentric contraction in this biceps flexion is when the movement is bringing the bar up. And then isotonic eccentric is when you maintain tension while the muscle elongates. So isotonic is same force, same amount of tone, but you're changing the length of the muscle. If it's shortening, it's concentric. If it's lengthening, it's eccentric. And then isometric is like your wall sit. You're holding it in one stable form for a long period of time, right? This demonstration like this. Make sense? This is really important for not just daily activity, but also postural responses as we go through our day, whether you're sitting, whether you're standing. And obviously these muscles fatigue. And it may be difficult for you to appreciate fatigue of postural muscles, but as you work with older patients, an older population, like a geriatric patient population, this will become much more apparent. Okay, well, they'll get fatigued sitting up. They'll definitely get fatigued walking or doing chores or cooking in the kitchen. We're going to talk about energy requirements, and I think this will be the last little section that we cover today. Okay, energy requirements. And this is a nice little place to hit the pause button, and I'll give you your code. So where do we get the ATP from? Well, we really have two main sources. We've got anaerobic fermentation, or we have aerobic respiration. And this is a sort of a review over Unit 1, where we talked about glycolysis being fast sources of energy, making a net of 2 ATP. And we talked about aerobic in the presence of oxygen being able to go through the citric acid cycle and the electron transport chain and generate more ATP, but it takes longer. So now let's get into the who cares. Like, why does this even matter? So there's a little bit of that exercise physiology that I was telling you that we were going to have. So we have the ability for some immediate demand of energy that are less than 10 seconds. There's a molecule in muscles known as myoglobin that actually houses a little bit of oxygen and can make it readily available really quickly. But it's a short burst. But beyond that, we have, more importantly, phosphate donations that happen within the muscle themselves from donating phosphate from molecules like creatine phosphate. So you can see in the muscle, we can actually utilize an enzyme known as myokinase, and we can actually steal phosphate groups from ADP molecules that have 2, adenosine diphosphate, borrow it, add it to another ADP, and make an ATP. You also have, through creatine phosphate, creatine kinase, which creatine phosphate will donate a phosphate, become creatine. This phosphate binds to this adenosine diphosphate using creatine kinase and makes ATP. These are less than 10 seconds, short bursts of energy. What about a little bit beyond that? Short term. Anaerobic. This is glycolysis using the glycogen lactic acid system. And you're looking at activities that are beyond 10 seconds, but you're at about 30 to 40 seconds worth of energy. Really no more than a minute. Some textbooks, some authors, some papers will describe the 59-second mark. Okay, for purposes of our class, let's say 30 to 40 seconds worth of energy. Definitely less than a minute, but it's beyond 10 seconds. And this is actually utilizing glycolysis. So these are going to be any sort of sporting event that's less than a minute, like a sprint. Okay, one of my kids is a hurdler, and she had a meet today, and my wife sent me the video because I couldn't make it today in the middle of the day. But she does the 100 and the 300 hurdles. And the 100 hurdles is less than a minute, and so it's all creatine phosphate and glycolysis. And so I've gone over these slides with her to explain to her why day of, like, fast sugar is so important, like 15, 20 minutes before her 100-meter hurdles. Now, the 300-meter hurdles is longer than a minute, so she's going to burn out of that energy. And after 40 seconds, you start using oxygen, using aerobic respiration through the electron transport chain. Okay? So 3 phases of energy utilization. Okay. For your bone